Okay, so since it's already 3 o'clock, let's get started with the session. Once again, I'll just keep my laser pointer. So yeah, welcome everyone to week 7 of the TA session for the course Bioengineering and Interface with Biology and Medicine. And I am once again to introduce myself. I am Arjun, and I am one of the TAs for this course for the iteration of January to April 2025. So we are in week 7 of this course, and this means that including excluding this week, there is only one more week of course material remaining. So in this week, what we are going to do is, as usual, we are going to go over a general recap of what was taught during the two past session in week 7 of the lectures, and then we will go over to solve some sample problems for week 7. Following that, we will, so since we are running behind time to discuss the assignment solutions for the week 5 and week 5 to week 8, this week I'm planning to cover both week 5 and week 6. So we'll be having doubt sessions in between them while we are solving again and again. So without wasting time, let's get started. So in week 7 is a continuation of week 6. So in week 6 we left off at evolution, and in week 7 we start in evolution. We also saw about the basics of bacteria and viruses, and some of the structural and functional components of bacteria and viruses. So once those facts are established, that virus, most of them have a protein coat and most of them function through proteins, the week 7's lecture starts with the introduction of amino acids and proteins, and we come to know that the amino acids are the building blocks of proteins, and proteins are large chain polypeptide molecules of amino acids, polymers of amino acids, which have which have several functions in our body, ranging from structure and mechanics, transport, channel formation of channels and pumps, and acid and base balance, fluid balance, formation of antibodies, those are also part of proteins, formation of enzymes which helps or catalyze key reactions in your body, production of hormones and signaling molecules. So proteins functions are not limited in our body, and they are and in our body there are like uncountable number of proteins. For like, as of now, there have been 200,000 plus kinds of proteins in our body, which uh, perform day-to-day functions in our life, which help us sustain life. So amino acids, once again to become, and those are like 20 kinds of amino acids. So we also saw the different kinds of amino acids, like aliphatic amino acids and aromatic amino acids, and we also saw how amino acids are formed. So amino acids basically have a carbon atom in the center, and to the carbon atom, amino group and a carboxylate group are attached, and on the other two sides of the carbon atom there is a hydrogen atom, and on the other side there is a R group. So the R group can be anything, and depending on the type of R group that is found, the amino acids are broadly classified into 20 different types of 20 different amino acids, essential amino acids. So we saw the structure, we saw how amino acids generally are, how amino acids generally have a amino group and a carboxylate group, and then we saw how these amino group and carboxylate group carry different charges in different medium. So when we go from acidic to basic medium, these will have positive and negative charges on each other, positive and negative charges on each other respectively, and in many cases, when it's dissolved or dispersed in a neutral solution, these exist in a sweeter ionic form. So this will form NH3 plus, and this will form as COO minus, which will be the most stable form of an amino acid, and which will be the most neutral form of an amino acid. And after that, we also saw about the definition of a peptide bond, how a peptide bond forms. So basically, when we have a amino acid 1 and amino acid 2, which have the amino group and the carboxylic group conjugated on each side of the carbon atom, so the peptide bond forms between the carboxylate ion on the carboxylate group of the first amino acid and the amino group of the second amino acid. So what happens is, so both of these groups interact with each other, and and upon subsequent leaving of a water molecule, we get this peptide bond, which wherein the C double bond O NH is formed. So the bond between C and N is called as a peptide bond, and the resulting molecule is called as a dipeptide molecule. And when further amino acids keep conjugating to the either the N terminal or the C terminal of the dipeptide, these form polypeptide molecules. So when a third uh, a third amino acid comes and conjugates to the carboxylate group through the third amino acid's amine group, then the resulting molecule will form a tripeptide, and so on and so forth. So multiple amino acids chained together will form a polypeptide, and so on and so forth. They form a kind of protein. So and when each peptide bond is formed, it results in the generation of a water molecule. And we saw about peptide bond, and we also saw how um, protein fun- and we also saw how protein have different functions. So any disruption we also saw about the central dogma, that how DNA is the DNA is the genetic storage, and how that is passed on to the subsequent generations, and how the DNA gets transcribed into mRNA, and how that mRNA gets translated into proteins through the and we also saw that amino acids have a 3-letter code and a single letter code, and we also saw that any disruption in this protein synthesis results in the generation of a misformed protein, or a protein which has a different sequence from the intended sequence, which is the main cause of several diseases or genetic abnormalities in our body. So any deviation from the any deviation or uh, any deviation or any mistake in the translation process results in the generation of a different kind of protein compared to the one which has a functional value in our body. So that will result in the disease condition. And and we also saw that um, amino acids are broadly classified into aliphatic amino acids and aromatic amino acids. And aliphatic amino acids mostly have hydrophobic groups, and they interact with each other through hydrophobic groups to form a hydrophobic core. And whereas on the other hand, aromatic amino acids have hydrophobic and hydrophilic groups. And we also saw how different aromatic amino acids are used for different functions, such as tryptophan and tyrosine, how they are used for protein estimation. And and in the subsequent part of the lecture, we also saw about the D form and the L form of the isomer, D form and the L form of amino acids, how they are the chiral form of each other. So chirality just means non-superimposable mirror images of each other. So when we take this D form, and if we take the mirror image of that D form, it results in the L form. But these two, a D form and L form, is not superimposable with each other. So these are chiral compounds, and we saw that except for glycine, all the amino acids possess this property of chirality. That is, their mirror images are non-superimposable over each other. And towards the last part of the lectures, we also saw about the structure of proteins, and we saw about the polypeptide chain and how that results in the alpha primary structure of the protein. So the primary structure of the protein is just laid out as such through the form of peptide bonds, where uh, each amino acid is conjugated to each other through the peptide bond. And we saw how this primary amino acids fold into each other in the form of alpha helix or a beta sheet to form the secondary structure of the protein. And then we also saw how the phi and psi angles in this. Once again, sorry, yeah. And we also saw how the phi and psi angles in these amino acid results in the formation of the it determines whether the subsequent amino acids will form an alpha helix or a beta sheet, and this phi and psi angles and the respective conformations are represented through the Ramachandran plot. And from a secondary structure, a protein can form into a tertiary structure, which is through hydrophobic and hydrophilic interactions. So the usually what happens is the hydrophobic groups get together with each other and then form a core, whereas the hydrophilic groups get on the outer surface of the protein. So this is called as the tertiary structure. And we saw that in most proteins, the most proteins have a structure called as a quaternary structure. So quaternary structure is the assembly of different tertiary structures which are non-covalently bound to each other through van der Waals force or by any other interaction. So on one hand, tertiary structure is bound with each other through hydrophilic interactions or covalent interactions through disulfide bonds. On the quaternary structure, they are different tertiary subunits are assembled with each other to form a quaternary structure through non-covalent interactions. So this is what we saw in the lectures regarding amino acids and proteins. So after that, we went on to discuss the course instructor Dr. Sanjeeva Srivastava went on to discuss about proteins and proteomics, and we saw that how different at different levels of the central dogma we can take out different components and then study them each. So for genes we have genomics, for mRNAs we have transcriptomics, and we have for proteins we have proteomics, and then proteins further differentiate into metabolomics and phenomics and harmonics and ionomics, and some of the proteomics also can be studied. Protein-protein interactions can also be used in the form of interactions, which forms an entire field called interactomics. So in proteins and proteomics, we initially saw how different gene sequencing projects were undertaken, with some of which include the Human Genome Project, and we also saw the different levels of genomics, transcriptomics, and proteomics, and we saw that how a single chain of DNA or a single chain of an mRNA, which contains different exons or introns. Exons are the coding regions, whereas the introns are the non-coding regions. We saw that how the single chain of DNA or the mRNA can form different types of protein through a process called alternative splicing. So alternative splicing is a process where the spliceosome binds together different exons in different combinations, which can be translated later through which can be translated later to form different proteins from a single mRNA. This thing. So the main component which used to undertake this alternative splicing phenomena is called as a spliceosome. And after spliceosome, we saw that how different proteins undergo different post-translational modifications. Here there are some there are some general summary of what are all the different types of post-translational modifications that can happen to a protein: phosphorylation, hydroxylation, methylation, acetylation, disulfide bonds, and ubiquitination and glycosylation. So these are the different types of post-translational modifications which can be this thing. And finally, we towards the end of the lectures we saw how a protein can be analyzed. So when we have a sample, there are different steps in which a protein can be analyzed. So the key steps involved in the protein analysis include this protein extraction, protein separation, protein identification, protein quantification, and protein characterization. So this will be the general summary of what will happen in a proteome analysis. And then we saw towards the end of the lectures what are all the ways in which we can separate protein. So protein extraction we saw in the previous weeks. How can we extract proteins from a given sample? In this week 7 lectures, we also saw how different proteins can be separated. So we saw different techniques like gel electrophoresis and two-dimensional gel electrophoresis, wherein in gel electrophoresis the proteins are only separated based on their molecular weight, but whereas in case of the two-dimensional gel electrophoresis, the proteins are first separated based on their isoelectric point through a process called as an isoelectric focusing. And after the isoelectric focusing is done, then the separated proteins are then further separated in another dimension based on their molecular weight. So this is called the two-dimensional gel electrophoresis. One dimension is the isoelectric point, and the other dimension is the molecular weight. And then after gel electrophoresis, we also saw how can proteins be identified and quantified. And we also saw techniques like MALDI and ESI. So MALDI stands for matrix dependent laser desorption ionization, where the sample of our choice, the protein of our choice, is coated in a matrix, and then it's bombarded with ions, or bombarded with the laser to produce ions, and which is then floated through a process, floated through an instrument called as a quadrupole analyzer or a time of flight analyzer, which is then used to uh, detect the proteins based on their charge to mass ratio. And ESI also follows a ESI, which is electrospray ionization, also follows a similar method of detection, but the production of ions differs compared to the MALDI process. And towards the end of the proteins and proteomics lecture, uh, we also got a gist of what it means by interactomics. So interactomics is a field which mainly focuses on protein-protein interactions. So we the course instructor also said that we can also do interactomic analysis through microarrays, which we saw in week 4 and week 5 of the lectures, where small drops of your samples can be plated in microarray plates, and their absorbance can tell us information about what is the concentration of the protein present in the microarray. Following that, towards the end of the lecture, we also got some demos on how to do two-dimensional electrophoresis and a process called DIGE, which is a differential gel electrophoresis. And we also saw in a process where in DIGE we can label different samples with different cyanine dyes, and then this uh, di- processes like DIGE helps us to avoid um, sample preparation or sample bias, and it also helps us to clear out things like sample positioning in when we are trying to image a sample. And in we also saw about techniques like chromatography tools, where we can do gel filtration and ion exchange chromatography and affinity chromatography. And towards the end of the week's lectures, we also got demo about the general bioinformatics tools that can be used to study any gene or protein of our interest. So we got introduced to this NCBI tool where we can uh, look where we can look for any gene or any protein or DNA or RNA sequence or information just by typing into the search bar if we know the ID, or if we just search for any of the gene or protein of interest we want, we can just search into this bar and then get the information. And we also got a demo of this blast, which is used for multiple sequence alignment, and which is not used technique used for multiple sequence alignment, which is just used to study the sequence homology and stuff. So there are different types of blast, but for the demo purposes we got an introduction into the nucleotide blast. And finally we got a demo of this software called cluster omega, which is used for multiple sequence alignment and to study the evolutionary relationships between those multiple sequences. So that's all the recap for this week 7's lectures. So we basically saw amino acids and proteins and their functions as well as their properties of amino acids. And then we went on to discuss about proteins, proteomics, and what are all the different processes that can be used to study, that can be used in proteomic analysis. And then we saw a recap, we got a demo of the basic bioinformatics tools. So I'll leave the room for like 5 to 10 minutes for a general doubt session. So if you guys have any doubts, you can shoot it up in the chat box. Okay, so since there are no doubts for now, we'll go on to discuss some sample problems from week 7. So we will get started. So question number 1 is, a team of researchers has purified two novel proteins A and B, which have lengths of 300 and 400 amino acids respectively. To study the intracellular localization of the proteins, they were expressed in a cancer cell line fused with two fluorescent tag proteins, GFP and RFP. So they have taken two proteins, protein A and protein B, and both of them, protein A has 300 amino acids, protein B has 400 amino acids, and to study them, they have fused protein A with GFP and protein B with RFP. So, and they have given the GFP and RFP weights and this thing as well. What are the approximate molecular weights of the fusion proteins? Given the average molecular weight of an amino acid in water are 110 Dalton. So, this question is pretty simple. It's just basic math. So what we do is, we just add the sum of the fusion protein will be, so they are asking what are the approximate molecular weights of the fusion proteins. So this is the total fusion protein. So the molecular weight of the fusion protein will be the sum of the protein A. So in the case of A, it will be sum of the protein A plus sum of the GFP. So GFP protein, this given here. The only thing is, you guys have to take into account that here they are given the molecular weight in Dalton, and here the molecular weight is in kilodalton. So kilodalton, 1 kilodalton is 1000 daltons. So how do you solve this is? So you just multiply the number of amino acids with the average molecular weight. So for protein A, it will be 300 into 110. So it will be around 33,000. Yeah, 33,000 plus 27,000 daltons, because it's 27 kilo for GFP molecular weight is 27 kilodaltons. So it's 27,000 plus 33,000. So for protein A, it's A fusion protein will be 60,000 daltons. So automatically all these options will not be correct, because only protein A here, only this option has protein A's weight as 60,000 daltons. So the correct answer will be A 60,000 daltons, B 69,000 daltons. So you can cross check this answer as well by checking it again. So for protein B, it will be 400 amino acids into 110 dalton, which will be around 44,000 daltons. Sorry, 400 into, yeah, sorry, yeah, 44,000 daltons. 44,000 plus 25,000, which will be 69,000 daltons. So the correct answer will be A 60,000 daltons, B 69,000 daltons. I'll try to attach a calculation of both the proteins' mass, and then I will upload it in the final Google Drive. This thing. I'll give you people 1 minute to go over the answer once again and do the calculation, and then we'll go to the next problem. Okay, so let's go to the next question. If you guys don't have any doubts. So this is the continuation of the first question. So as the protein A is tagged with GFP and protein B is tagged with RFP, then at which wavelengths would you like to visualize them under a confocal microscope? So uh, this question is pretty conceptual but pretty simple, I think. So they are just saying protein A is tagged with GFP and protein B is tagged with RFP. Then at which wavelength would you like to visualize them under a confocal microscope? So they are given options for A's wavelength and B's wavelength. So one way to go around this is just to have a general idea of how the UV visible spectrum will look like. So normally, if you want to image something in the UV visible spectrum, so you want to, so there is a GFP and RFP which are both visible under a microscope. So you would want to like use the visible range of the microscope from 400 to 700 nanometer in order to use them. So automatically this 260 and 280 nanometer which is under UV range will automatically not be used. So 260 and 280 will not be used. Similarly here we have 350 nanometer. This will not be used, and 350 for red fluorescent protein. Protein B, which is 350 for RFE, will definitely not be used. So since red is at the far end of the UV visible spectrum, red will be used at somewhere between 600 to 700 nanometer. So that's how we should take the calculation. So ideally, this option B, which is A will be visualized at 510 nanometer and B will be visualized at 688 nanometer, will be the ideal answer, because your red will most probably stop at your visible spectrum will most probably stop at around 700 nanometer. If you go at 810 nanometer and 988 nanometer, it will go to the IR range. So the correct answer will be A will be visualized at 510 nanometer and B will be visualized at 688 nanometer wavelength. So there are no doubts. I'll go to the next question. So this is also, I think, the continuation of the uh, previous questions. So upon visualization of both proteins using high resolution microscopy with the 100x magnification, both proteins were found in the peroxisome. They resembled the image depicted below. They please explain what does a yellow color signify when the expression of only green and red fluorescent tagged proteins were being monitored. So yeah, so what they are saying is, so I'll read the question again. Upon visualization of both proteins using high resolution microscopy, both proteins were found in the peroxisome. So this is the peroxisome, and both proteins were found. They resembled the image depicted below. Please explain what does a so we have a GFP filter where only the green things are present, and we have a RFP filter where only the red things are present. But if we fuse them both together in the merge filters, there is a there are green stuff, and there is yellow stuff. There are no red stuff. So they are asking us to explain what does the yellow color signify, because we only imaged both GFP and RFP. So this yellow color most likely what it is it is the combination of your red filter and your green filter. So when you merge them both, the regions where both GFP and RFP are expressed will give you the combined color, which is yellow in color. So the answer will be the yellow color signifies the co-localization of both proteins together in the peroxisome. So this is a multiple choice question only. So only one option can be correct. So it's better that you don't confuse yourself by taking these options into consideration, because only this option can be the most logical of them all. That both of them are like co-localized in the same region, so it gives you the yellow color. So I'll go on to the next question. A schematic of a peptide bond is shown below. They entail a particular kind of chemical bonding between amino acids and are the fundamental building blocks of proteins. Which of the following statements about peptide bonds is correct? So they have shown a picture of a peptide bond. We also discussed about peptide bond in the recap, where we have C double bond O NH, and then you have amino acids here, and then once again you are C double bond O NH here. So they are asking, they are given four statements and they are asking which one of them is like most correct. So option one is the 3D folding of proteins happens because of interactions between the R groups of amino acids with the carboxyl group in peptide bonds. Second option is peptide bonds are very flexible and can rotate freely in all directions. So third option is the formation of peptide bonds occurs through dehydration synthesis, wherein the amino group of one amino acid reacts with the carboxyl group of another amino acid to liberate a water molecule. Fourth option is peptide bonds are formed through hydrolysis, breaking the connection between carboxyl group and the amino group of adjacent amino acids. So out of all these options, only like, so we already knew that the peptide bond formation, it happens through the dehydration process, where the COOH and NH2 releases a water molecule and then they form the peptide bond. So this option 3 seems the most valid, and it happens to be happening all the time when a peptide bond forms. So the correct answer is this. So the formation of peptide bonds occurs through dehydration synthesis, wherein the amino group of one amino acid reacts with the carboxyl group of another amino acid to liberate a water molecule. So I'll move on to the next question. So using size exclusion chromatography, you have purified a 100 kilodalton protein molecular weight. So 100 kilodalton is the molecular weight of the protein. To check the quality of the purified protein, when you performed a page, so there is with the polyacrylamide gel electrophoresis, in the presence of SDS and beta mercaptophenol, it yielded two different bands, each having a molecular weight of 20 kilodalton and 30 kilodalton respectively. However, a native page gave you a 100 kilodalton band. To ensure reproducibility, the purified protein was loaded in triplicates every time. In both cases, a known protein marker was also run to track the molecular weight of the bands. Below is a representative of both the gels. What could be the best explanation of this scenario? So, uh, this is like pretty a thoughtful question. So they are running triplicates of the gel every time, and they are doing SDS page and native page. So native page you don't use SDS and beta mercaptophenol, whereas in SDS page you use this SDS and beta mercaptophenol. So if you know the functions of both of this, which was explained in the, if you know the function of both SDS as well as beta mercaptophenol, which was explained in the demo of this lecture, week 7 lectures, I think you will be able to solve this question. So they are used in SDS page. What happens is you are getting two bands, one of 30 kilodalton and one of 20 kilodalton, and in native page you are getting a single 100 kilodalton band. Yeah. So they are asking why this thing could happen. Why this thing could happen. So I so we have four options here. First option is the protein has three subunits, one having 100 kilodalton, one having 30 kilodalton, and the last one having a 20 kilodalton molecular weight. Second option is the protein is either a homotetramer of 20 kilodalton or a homotetramer of 20 kilodalton units. I think it's 30 kilodalton, but I think it's typed wrong. Third option is the SDS page was faulty, hence has multiple wrong bands. However, the native page, I think this option is also not complete. Fourth option is the protein homodimer of two heterodimers. The protein is a homodimer of two heterodimers, 20 kilodalton and 30 kilodalton. So, which one will it be? So, I'll give the room like 5 minutes to just go over both the pages and then let me know what the uh, probable answer is. Okay, so we'll start discussing the answer. So we'll go over the options one by one, and then we'll see which one of them makes more sense. So the first option is the protein has three subunits, one having 100 kilodalton, one having 30 kilodalton, and last one having 20 kilodalton molecular weight. So before going on the options, I'll also tell you like why do we use this beta mercaptadinol. So beta mercaptadinol is used to break the disulfide bonds between your subunits. So if you have a dimer or a trimer or a tetramer, this beta mercaptan, it's formed together due to the this thing beta mercaptanol, and SDS is a surfactant. So what these two things do together is it helps you to break the disulfide bonds, and then it used to separate the fragments nicely. So if you have, let's just say that we have three subunits, one have 100, 30 and 20 kilodalton. The native page which doesn't use any of the surfactants or the disulfide bond breakers, it should have, it will generally show you the total molecular weight of the protein. So if it is, if first option is correct, then your total molecular weight should be 100 plus 30 plus 20, which is like 150 kilodalton. But we are not getting any band here at 150 kilodalton. We are only getting the maximum band at 100 kilodalton. So this is not a correct option. Second option, protein is either a homotetramer. Tetramer means 4 units of 20 kilodalton or a homotetramer of 30 kilodalton units each. So if it's a homotetramer of 20 kilodalton, then the total, once again, the total molecular weight should be 20 into 4, which is 80. Or if it's a homotetramer of 20 kilodalton, it should be 30 into 4, which is 120. But we don't have any band at 80 or at 120 kilodalton as well. So this option is also wrong. The SDS page was faulty, hence has multiple wrong bands. However, the native page was not faulty. Then this is not possible because your if SDS page is wrong, then you could have had irregular bands as well. You have literally done triplicate analysis, and all three of them have bands at the same place. So it may not be correct that the SDS page is faulty. But we'll come to the last option. The protein is a homodimer of two heterodimers, 20 kilodalton and 30 kilodalton. So what this option is saying that there are two homodimers and each one is a heterodimer where one unit is 20 kilodalton and one unit is 30 kilodalton. So in this case, the total weight will be 20 plus 30 plus 20 plus 30, which is 100. So the native protein, it will give you 100, it will give you this thing 100 kilodalton. And if we break this, we will get two proteins of 20 kilodalton and two proteins of 30 kilodalton. And those are what we are getting it here. We still don't know what proteins are there in each 20 kilodalton, because then we have to coat our antibody as well. But for now, we are getting bands at 20 kilodalton and 30 kilodalton. So the correct option is the protein is a homodimer of two heterodimers, 20 kilodalton and 30 kilodalton. So I'll go to the next question. We'll come back to this if we have any other doubts. So a synthetic peptide contains 4 negatively charged and 8 positively charged amino acids at pH 6.1. When the peptides are treated with an alkaline solution having a pH of 8.9, 4 positively charged groups become deprotonated. At pH 8.9, What is the net charge on the peptide? What will be the direction of movement towards cathode or anode if the alkali treated peptides are now placed in an electrical field? So I'll read the question once again. So we have a synthetic peptide. It has 4 negatively charged and 8 positively charged amino acids at 6.1. So 6.1 is a pretty uh, mildly acidic pH. So when the peptides are treated with an alkaline solution having a pH of 8.9, 4 positively charged groups become deprotonated. So now at pH 8.9 we have 4 negatively charged and 4 positively charged amino acids. At pH 8.9, what is the net charge on the peptide? So if the and what will be the direction of movement if the alkali treated peptides are now placed in analytical field? So at pH 8.9 where we know that there are 4 negatively charged and 4 positively charged amino acids because the other 4 positively charged amino acids are deprotonated. So the net charge will be minus 4 plus 4, which will be 0. So if a charge on the peptide is 0, then it will not move towards cathode or anode. So the movement of the peptide will not happen. So the correct answer is 0. Movement of the peptide will not happen. So I'll go to the next question. The arrangement of many polypeptide chains or subunits into a functional multi-subunit complex defines a protein's quaternary structure. Which of the following statements about the quaternary structure of a protein is accurate? So the option 1 is the beta chain direction in a protein's primary structure is the only factor that determines its quaternary structure. Regardless of size or purpose, all proteins have their quaternary structure. Quaternary structure involves the non-covalent interactions and spatial arrangement of multiple polypeptide chains. Quaternary structure is only relevant when it comes to enzymes and has no role in structural or transport proteins. So this is a bit theoretical question. The answer is it's non-covalent interactions. Quaternary structure involves non-covalent interactions and arrangement of multiple polypeptide chains. So I'll go to the next question. The differential in-gel electrophoresis or DIGE process is illustrated in the following diagram, which uses two distinct tissue sample types, normal and cancerous tissues, N, C, and a combination of both N plus C have been combined with Psi2, Psi3, and Psi5 cyanide dyes. So we have normal cell lines which are coated with Psi2, cancerous tissue sample which is coated with Psi3, and uh, the combination of normal and cancer which is coated with Psi5. So according to cyanide dyes, the gel image was scanned for three distinct wavelengths following electrophoresis. Select the right statement. So what will be the most correct statement? So the options are gel N plus C always has fewer spots than gel N and C together. The N plus C gel excludes the probability of batch to batch variation and it's considered to be the good reference for further data analysis. The proteins in gel N and gel N and gel C are entirely different. Both A and B are correct options. So we'll go over one option, all the options. So gel N plus C always has fewer spots than gel N and C together. So this is not completely true because gel N and gel C, the normal cancerous tissues and normal tissues and cancerous tissues will have proteins. Some of the proteins will be common to each other, well, some other proteins will not be common to each other. So the fact that, uh, but the combination of them will represent all the proteins which are there in both the samples. So N plus C will always not have fewer spots than gels N and C together. It will have either equal number of spots, or yeah, it will have equal number of spots. So that option is not correct. So second option, the N plus C gel excludes the probability of batch to batch variation and is considered to be a good reference for further data analysis. This is true because that's why we use this dye, because we mix all the samples with different cyanine dyes so that we can avoid batch to batch variation with respect to gel analysis. Third option, the proteins in gel N and gel C are entirely different. Once again, we discussed that they can't be entirely different. Some proteins will be common to both normal and cancerous tissues. So the correct option is option B. The N plus C gel excludes the probability of batch to batch variation and is considered to be a good reference for further data analysis. I'll try to. I'll go to the next question. So you have identified an unknown protein having a molecular weight 23 kilodalton from mass spectrometry based proteomics that plays a crucial role in cervical cancer tumorigenesis. You have further sequenced the protein to get its amino acid sequences. Now you wish to study the conserved regions of the protein in detail. With what bioinformatics tools will you use to understand about such crucial information about the protein? So this is also a pretty theoretical question which we saw in the final bioinformatics part. So if you want to know, if you have a sequence with you and if you want to find similar sequences, then you normally run this nucleotide blast, or yeah, or you basically run blast, nucleotide blast or protein blast. In this case, we have the amino acid sequences, so you run protein blast, and then after getting the protein blast, you will get the other other sequences which are common, or which are matching some percentage to your sequence of interest. So you will get those sequences as well, and then you will perform multiple sequence alignment using cluster omega to look for the conserved regions or common regions in the protein. So the correct answer, so that in cluster omega you will also see which regions are deleted and which regions are added or modified. So the correct answer is run a blast to get similar sequences from different organisms and then perform multiple sequence alignment to look for the conserved regions in the protein. So I'll go to the last this thing. If you, I'll be uploading this recording to YouTube, so you can pause it and also look at this thing. So you have identified a novel drug that can bind to an oncoprotein named SRSF3. How will you visualize the protein drug interaction and gain more idea about the binding site and its interacting amino acids? So I think this is a pretty complex question. So this you will learn in the next week's lectures. But the basic thing is, if you want to study the protein drug interaction, you might have to use tools like PyMol to perform molecular docking. So first you will perform a molecular docking between your binding site, and you have to visualize using PyMol. So that's all of the sample problems for this week. This week's questions. So I'll have a short doubt session for the questions and the concepts discussed in week 7. Till now it's 3:55, so we'll probably have it till 4. So after that, we'll discuss some of the assignment questions from the previous weeks, especially week 5 and 6. Okay, so we'll go and discuss this thing. So let's get started with this. So first we'll discuss the week five's. First we'll discuss the week five's answers. So yeah. So first question is Ramesh was provided with specimens of gram negative and gram positive bacteria. After performing the steps of gram staining, he proceeded for observation under a microscope, but he saw both the slides purple in color. What could be the possible reason for this? So this is also a pretty straightforward question in that assignment. So your gram staining is purple in color, but if you want to observe gram negative and gram positive, you need to also add the counter staining, which is saffron. So if we had forgot to perform the saffron staining, which is the counter staining, then he would have seen everything purple. So the correct answer is he forgot to perform saffron and staining. So this question is pretty straightforward. So question number two is, suppose you are screening for an antibiotic which works by inhibiting peptidoglycan of cell wall. You take two bacterial strains A and B. You notice that A stains violet in gram staining and B stains red. Antibiotic will be effective against which of the following strains? Strain A, strain B, both A and B cannot be determined with gram staining experiment. So there's also a pretty straightforward question about this thing. It's strain A. So how does this strain A is? So we know that gram staining is used to identify gram positive and gram negative, and the gram positiveness is about the presence of the peptidoglycan cell wall, and the antibiotic which is given in the question, it works by inhibiting the peptidoglycan of cell wall. So let the cell won't this thing. So we are noticing that A stains violet in gram staining and B stains red. So A is automatically gram positive and B is automatically gram negative. So A has the peptidoglycan cell wall. So if you are adding an antibiotic which inhibits the peptidoglycan, A which has the peptidoglycan cell wall will be the antibiotic will be effective against strain A. So the correct answer is strain A. So, third question is, which of the following conditions led to the death of the mouse in the famous Frederick Griffiths experiment? So, they are given four these things, living four statements. So, we have to find out which conditions lead to the death of the mouse. So, first statement is living estrain bacteria injected in mouse, living R cells injected in mouse, heat killed S cells injected in mouse, mixture of heat killed S cells and live R cells injected in mouse. So, this is also a pretty straightforward question. It's 1 and 4 because S strain is the virulent strain which will kill the mouse, and R strain is the non-virulent version. If heat killed virulent strains are injected, the mouse won't die. But if heat killed virulent strains and non-virulent strains are subsequently injected, the genetic material gets of the virulent strain gets transferred to the non-virulent strain, which results in replication of the virulency, which results in the death of the mouse. So the answer is living S strain and mixture of heat killed S cells and live R cells. So I'll go on to the next question. Cinnabar eyes are a recessive sex-linked trait in fruit flies. What? So cinnabar eyes are a recessive sex-linked trait in fruit flies. What percentage of F1 males will have cinnabar eyes if a female with cinnabar eyes is crossed with a wild-type male? So they are asking what percentage of F1 males will have yeah, what percentage of F1 males will have cinnabar eyes if a female with cinnabar eyes, which is a sex-linked trait, is crossed with a wild-type male? So if sex-linked recessive means the female should have. So I'll just take a pen. Right. Yeah. So keyword is recessive sex-linked trait. So if the female, this question, if a female has cinnabar then she should have XC, XC. So both of them should be, uh, both the alleles should be with cinnabar specific allele, so that she will be having the cinnabar. And then wild type male is normal, so it will be, he will be XY. So they are asking what percentage of F1 males less cinnabar. So if both the alleles are this thing, then definitely the male will have the cinnabar because let's just say if a wild type male has XY, so regardless of which allele crosses with the Y of the male, the since it's a recessive sex-linked trait, the male is going to get the cinnabar. So 100% is the answer of F1 males. So I'll go to the next question. Mahesh is suffering from a sex-linked recessive disorder, hemophilia. He has a daughter who has a normal phenotype. She marries to a man who is of normal phenotype, but her mother-in-law is a carrier of hemophilia and her father-in-law is suffering from the disease. What will be the probability of their daughter being hemophilic? So I'll give this a bit of time to solve this answer. Meanwhile, I'll just, yes. So we'll just proceed to solve the answer. So once again, so just try drawing a general thing of how what is the case. So Mahesh is suffering from hemophilia. So let's just say Mahesh is XY and it's a sex-linked recessive disorder, hemophilia. So it's on the X chromosome. Sorry, there was a small network issue which my network connection got disrupted. I'll share the screen once again. Uh, just a second. Yes. Sorry for the small disturbance. So we will continue with solving the question. I'll just load my point pen. Yeah. So this is let's just say Mahesh has hemophilia. So he has XH and Y. So Mahesh's daughter. So Mahesh's daughter, she has a normal phenotype. So let's just say she has. So she is not even a carrier. She has a normal phenotype XX. So Mahesh Mahesh's daughter marries to a man who is of a normal phenotype. So this is XY, but her mother-in-law, that is the husband's mother, is a carrier of hemophilia. So if she is a carrier, then we'll denote it as XH and X, and then her father-in-law is suffering from the disease. So we'll denote it by XH Y. Yeah. So what would be the probability of their daughter having hemophilic? So father is hemophilic, but father won't pass his. So we know for a universal fact that father won't pass it to their sons. Father will only pass it to their daughters. And uh, if mother has hemophilia, so chances are that and they are saying that the son is normal. So the son is not getting the hemophilic allele of the mother. Since mother is a carrier of hemophilia, she has a hemophilic allele and a normal allele. So if son is normal and is not even hemophilic, uh, he would have not got the hemophilic allele from the mother. Instead, he would have got the normal allele from the mother. So he is perfectly normal XY individual. So they are asking what would be the probability of their daughter being hemophilic. So only the father can pass the disease to the daughter. So if the father doesn't have hemophilia, then the daughter also cannot have hemophilia. So the answer is 0. So I'll repeat the answer once again. So yeah, so I'll repeat the solution of the problem once again. So let's just assume that this is Mahesh. So Mahesh is having hemophilia, and hemophilia is on the X chromosome. So he will let's just represent Mahesh's phenotype as XY, where XH is the hemophilic allele, and Mahesh has a daughter who has a normal phenotype. So she has XX because Mahesh cannot passes. Mahesh did not passes thing to his daughter. Oh, sorry. So actually, yeah. So actually, I made a mistake here. So the father would have passed down the hemophilic allele to her daughter. So we'll represent one of the things as H. But still the answer stands as being 0. So the daughters the daughter will have a normal phenotype, but the genotype will be XHX. So that she'll be a carrier of the disease and not a affected individual. So she is marrying a person who is completely normal phenotype, so that is XY, but her mother-in-law is a carrier and her father-in-law is suffering from the disease. So let's just say the husband's father is having XHY, where XH is the affected allele, and let's take the mother-in-law's allele as XHX, so that she can be a carrier, so that only one of the alleles is hemophilic. Since it's a recessive disorder, she won't be affected by this and she will be a carrier. And mother, yeah, so she has passed down the allele to her son, who is a normal phenotype, so he is XY. So now they are asking what will be the probability of their daughter being hemophilic? So we know for a fact that fathers will only transmit the disease to the daughter. So if the father is perfectly normal, then the daughter will also be perfectly normal, but she can be a carrier or a normal individual. She can either get the normal allele or the hemophilic allele, in both the cases the daughter will not be hemophilic. So the probability will be 0. So I'll move on to the next question if we don't have any doubts. So given below is the representation of a normal chromosome in which letters represent the genes and dot represents the centromere of the chromosome. Okay, so they are represented a chromosome in which dot is the centromere, and then on both sides of the centromere we have genes. Which of the following is the correct match of the given chromosome with their respective structural alterations? So they are asking which one of them is a correct structural alteration. So here, he in this first case we are having PQRS. So T is missing, and instead of T, they are having a misplaced G. So the answer is only one, because this is the ideal structural alteration, because there is a additional base to this part. So I'll go to the next question. Let's assume a bacterial cell which is initially growing and replicating in 14N containing media is transferred to 15N containing media. What will be the composition of DNA of bacterium after two generations in 15N medium? So this question is also like pretty straightforward from the Michelson and Stark experiment. So the correct answer is 15 and 15N will be 50% and 15 and 14N will be 50%, because the replication happens in two cycles. In the first cycle, all the bacterium will have 15 and 14 and hybrid DNA, and in the second cycle, we will have two 15 and 15 and DNA and two 14, 15 and 14 and DNA, which will be 50% and 50%. This is like the opposite of Michelson and Stahl, where uh, the original Michelson and Stahl experiment, we have tagged 15 and bacteria, and then we have grown them in 14 and media. So here we are growing 14 in media and then we are transferring it to heavy media. So it's just the opposite, but still the same outcomes are happening in the second cycle. After this, it will change. And in the Michelson and Star experiment, we would have had, instead of 15 and 15, we would have had 14 and 14N as 50%. But since we are transferring it from 14N to 15N, we will be having 15 and 15N as 50%. So we'll move to the next question. A syndrome is crossed. A syndrome is caused when a group of symptoms occur together and characterize a particular abnormality or condition. Turner syndrome is a genetic disorder caused by abnormal cell division. What chromosomal abnormality is the cause of the disease? So this is also theoretical. So the answer is trisomy of X chromosome causes Turner syndrome. So all the others cause Down syndrome, Pato syndrome, and yeah. So I will go on to the next question. So Rashmi was initially growing bacteria in 15N media, and then she moved them to the 14N media. Let us assume that DNA replication follows conservative model. Then which of the following bands would appear after density gradient centrifugation after two generations in 14N medium? So conservative approach is, so we had three models of DNA, right? Conservative, semi-conservative, and mixed. So in conservative, the parent chains will remain intact. The newer formed chains will form new DNA. So even in the conservative model, we will have, so in conservative in conservative model, what will happen is if 215 and DNA are in, let's just take a semi-conservative model for example. So if here 215 and strands, both of them will separate for replication, and each 15 and strand will have 14 and 14. So we'll have 15 and 14 and 15 and 14. I can also draw it and show it to you. So in semi-conservative model, we will have in semi-conservative model we will have 215N, and after replication it will form two different strands, and it will have 15N and 14N. This is the semi-conservative model. But in the case of your conservative model, if two strands are there, the parent strand remains the same, whereas your new 14N strands form separately. So this is the first cycle, and after the second cycle, it will continue to divide. So this will remain the same. This will be the parental strands. This will generate one strand. This will generate one strand, and both of them will be this thing. So I can also draw the thing once again. So yeah, so this is the difference between your semi-conservative and your conservative model of DNA. So in semi-conservative model of DNA, after two cycles, you will have two hybrid 15 and 14 strands, and you will have two 14 strands. And but in the conservative model, you will have only one 15 strands and three 14 strands. So in this question, they are asking which of the following bands will appear after density gradient centrifugation after two generations. So we will be having one band of heavy DNA and one band of not medium density with yeah, this will be the answer. One band of light DNA and one band of DNA with heavy density. So we'll be having only one band for 15N and one band for 14N, which is the heavy and light forms. We will not be having any hybrid medium density chains like in the semi-conservative model. Yeah, I'll just show the answer here for now. Then later I can. So I'll just stay in this window for a couple of time just for you guys to understand the question. I'm sorry the ink is over the correct answer, but yeah. So we'll move on to the last question. I'll give me this pointer. So brown wings in Drosophila is dominant to white wings. B. Red eyes color is dominant to white eyes color. What fraction of the progeny of the cross, capital B small b, capital R small r cross, capital B small b, capital R small r will have brown wing and white eyes? So this is also pretty simple. If you do the math, you will get the answer as 1 by 2. So I'll leave a short down session for all the answers how we discussed in uh, week 5 answers till like probably 4:30, and then we'll go on to discuss week 6 answers. So for solving the 10th question, you just need to look at the, you can either use the probability theory, or you can just look at the uh, brown wings and eye characteristics separately. So if you take only the wing characteristics in capital B small b and capital B capital B, regardless of however you cross them, you are going to get only brown wings for the cross. So brown wings is going to be probably 100%. So you just need to see what fraction will have brown wing and white eyes and brown wing and other red eyes. So that lies in this question. So capital R small r is a case of this cross, which will give you half red and half dominant and half recessive, which is half red and half white. So your brown wing and white eyes will finally have half probability. Brown is confirmed. Like brown will every all the progeny of the cross will have only brown wings. Whether you have the white eye or the red eye will be the question, which will be half and half because the eye color is a classic case of a test cross. So, okay, we can also have the final doubt session in the end as well. So we'll go first finish the questions for the week 6 assignment. So week 6 question number 1: DNA gets compressed into a smaller volume inside the nucleus, finally forming the nucleosome structure. The nucleosome is the fundamental subunit of chromatin. Select the correct statements about nucleosomes from the image below. So they have given nucleosome wrapping around the DNA, and they have given four options about what those are. So we just need to answer what each of them will be. So this is also a very pretty theoretical question. So correct answer will be A is histone modifications the whatever modifications that are done to a histone. B will be the core histone proteins. I'll just move my camera. B will be your core histone proteins, which are wrapped around your DNA, and C is DNA wrapped 1.47 times around nucleosome. This is a very theoretical question which you can find the answer for in the Campbell biology book. So I'll go to the next question. Yeah. So assume that you are studying cell division in a metastatic cancer cell line. Your senior gave you a subline of cells where she fused kinetochore proteins with GFP, a protein that produces green fluorescence. After reaching confluency is a process where your cells cover the entire area of growing. So you harvested the cells and mounted them on a slide. Which part of the cells do you think will show fluorescence? If you harvest the cells at their logarithmic growth phase followed by cell lysis and cell centrifugation, then what part of the solution will contain most of the green fluorescence? So this is also a pretty theoretical question. The answer is the initial part will only centromere will this thing show your fluorescence because you are conjugating your kinetochore. And if you centrifuge it, most of the pellet will contain most of the fluorescence since your pellet has the cells. So I'll move on to the next question. Rapidly proliferating cells may begin to appear aberrant and disordered as they continue to undergo change, genetic mutation. This stage of a transforming cell can be described best by which of the following option. Uncontrolled cell division, on the other hand, may result in an overabundance of cells that appear normal under a microscope. What do you call these conditions? So this also is a normal theoretical question. The answer is dysplasia and hyperproliferation. So dysplasia is a process where um, dysplasia is a process where cells rapidly proliferate and they appear disordered, and hyperproliferation is the other uh, process where they happen to be overabundant but appear normal under a microscope. So we'll move on to the next question. The graphics below depicts the sequence of events that occur throughout the development of an embryo. Select the appropriate answer for 1, 2, 3 and 4 from the below mentioned options. So this question we already saw in last week's sample problems. So the answers is 1 represents fertilization where the sperm and the egg fuse together to form zygote. The second process is cleavage, where the zygote undergoes two-cell stage and a two-cell stage and a four-cell stage and the eight-cell stage. And the third process is gastrulation, where the blastulation, where the blastula is formed. And the fourth process is gastrulation, where the blastula divides further to form the embryo. So and the gastrula also has a germinal layer such as your ectoderm, endoderm, and mesoderm. So, fifth question, pick the incorrect statements about the gastrulation event. Gastrulation is the stage in the early embryonic development where the blastula is reorganized into a multi-layer structure known as the gastrula. During gastrulation, the early embryo undergoes a lot of activity to establish cell polarity. Uh, Early embryo undergoes a lot of activity to establish cell polarity. By the end of gastrulation, the embryo initiates to differentiate and generate discrete cell lineages and build the body's essential axes. So out of this, gastrulation is last question we saw. So gastrulation is a process where the multi-layered structure known as gastrula is formed, where you also have the germinal layers. And third option, by the end of gastrulation, the body's axis is also formed and the cell lineages are also determined. So the only incorrect option here is 2, where the early embryo undergoes a lot of activity to establish cell polarity. Because the cell polarity is established in the preliminary stages itself, where the animal pole and vegetal pole are determined, the polarity of the cells, and the cell, the sperm also binds to the region where the animal pole is, sorry, the vegetal pole is present. So, sixth question, SARS-CoV-2 infects humans via the ACE2 receptor found on the host cell surface. Let's assume that the entire glycoprotein is encoded by one gene. What is the maximum number of genetic variants that could exist in Asian population for that gene? So this is like kind of a theoretical this thing. Any number of genetic variants can exist for a given gene. Even small changes present in the gene can lead to multiple this thing. So the answer is unlimited. So seventh question. So this is a mathematical problem. So an autosomal recessive illness exists in a community that is also thought to be in Hardy-Weinberg equilibrium. The condition affects so affects 16 out of every 100 people. What are the diseases allele frequencies in the population? Given that M is the dominant allele and capital M is the dominant allele and small m is the recessive allele. Also, what should not be done by the individuals in the group to keep the population in Hardy-Weinberg equilibrium? Choose the best combination. So the answer, so so technically the answer is pretty easy. So we all know the conditions where Hardy-Weinberg equilibrium can be established, and all the options, in the second options, this option, the individual should not migrate to other places, holds true, because the other options do not hold true under Hardy-Weinberg. If they are undertaken, Hardy-Weinberg equilibrium cannot be established. So technically, this option will be the most correct option. But you can also solve it mathematically to see how this can be solved. I'll give you guys like 4 minutes till like 4:40 or something to solve this. I have already given you a hint that the first option is the right one, but you still need to verify it mathematically how to solve this. Okay, so I'll just proceed to solve this. So I'll just take a pen. Yeah. So keywords here in this question are autosomal recessive illness, and then this statistic that 16 out of every 100 people are affected. So this is the so so autosomal recessive disease means this condition is M, the small m, and the capital M is the normal dominant allele, which is the normal condition, not diseased people. It can be either homozygous dominant or heterozygous dominant. So they are saying that the condition affects 16 out of every 100 people. So we need to find out the homozygous recessive frequency, which is Q. So what will be the Q? Q will be the homozygous recessive frequency, which is the total number of recessive people affected divided by the population. Sorry, Q square, sorry, which will be 0.16. So the allele frequency is Q square, which is 16 divided by 100, which is 0.16. So what will be Q here? Q will be the square root of Q square, which will be 0.4, which is the square root of 0.16 is 0.4. So we know which is basically your small m, which is the allele frequency. So if you know m, so capital M is according to Hardy-Weinberg equilibrium. Capital M is easy to get because it's just 1 minus the recessive allele frequency, which is your 0.6. So I'll anyways put the answer as well. So capital M is 0.6 and small m is 0.4, and the individual should not migrate to other places to maintain Hardy-Weinberg equilibrium. If this question is clear, I'll move on to the next question. Yeah. So the graphics below depict two very distinct forms of evolution. Correctly identify them using the right logic. So we have two different this thing, 1 and 2. So one of the in the first graphic we have sugar glider from Australia and the flying squirrel from North America. Both of them have the same kind of this thing. So we have to find out whether they are convergent evolution, divergent evolution, or parallel evolution. So this one is pretty easy. So this is the example of human, cat, and whale. We are just seen according to the environment, all the same thing as the diverged into different this thing. So this we know that 2 is a example of your divergent evolution, and 1 is, so using that to solve 1 as a convergent evolution, because in 2 you have a common ancestor and the common ancestor has had the same thing. So and depending on your environment, the same bone structure have modified into different parts for different purposes. So correct option is 1, convergent evolution, in which both species exhibit comparable features while not sharing a common ancestor, and 2, divergent evolution, in which groups of the same progenitor led to speciation while keeping structural similarity. So I'll go on to the next question. In the laboratory, we are preparing for a cloning experiment that will necessitate embryonic stem cells. Choose the correct combination of embryonic stem cell phases. So we also saw in the cloning this thing that if we have to use embryonic stem cells, they should be very early. So option 1, they are unsuitable for cloning studies. Option 2, they can be easily extracted from the blastula stage. Option 3, these cells can multiply endlessly in specific culture media. 4, they are highly specialized cells. So which one of them? So the correct answer will be 2 and 3, because if I already told you that if they are highly specialized cells, they can't differentiate into whatever cells you want. So you can't use this type of cells. And if they are unsuitable for cloning studies, then why will you use it for cloning? Because the main part of this thing is to use for cloning. So if you are extracting a stem cell to be used for cloning, you will extract a very early stage stem cell, especially from the blastula stage, and they should be able to multiply for a specific amount of time. Endlessly is preferred. So the correct option will be 2 and 3. So the last question. So homologous organs are those which are shared by several species and are comparable to those of other species. Which of the following evolution patterns reflects the presence of these organs? So they are saying the question is about homologous organs. On the other hand, one one unique phenomenon observed in nature quite frequently is that variation in gene frequencies within a population can arise by chance rather than natural selection. Pick the correct phenomena as well. So this is also a very theoretical question. So first one is the your divergent evolution. Second one is your genetic drift. So divergent evolution is where you have a common ancestor, like we saw in the previous two questions, and depending on the function and environment you are present in, your structure or your phenotype will be altered to suit the specific environment. So that's all the this thing for this week. So I'll leave the remainder of the session for a doubt session, or since no one is there in the room right now, I'll end the session right now. So the next session for next session, that is the eighth week of this TA session, will be held next Saturday, 15th March, from 3 p.m. to 5 p.m.